Great. So it is 1:30. I think we'll get started right away. And I realize I've moved along to my second slide. But hi, everyone. I'd just like to welcome you to our 5th session of the Summit Speaker Series, which really provides an opportunity for our network to come together to listen to talks by our network investigators and our stakeholders and trainees in our network. And so today I'm very pleased to welcome the speakers of this session and to introduce the topic. And so today's topic will be engineered biovesicles, a new platform for cancer immunotherapeutics. And with that being said, before we jump into it, we do have a session, and our next session event will be taking place on March 10th from 2 o'clock to 3 o'clock p.m. And it will be focusing on what it takes to manufacture a vaccine from a biomanufacturing perspective. And this session will be oriented to everyone, but it will be focusing, I think, on a talk that's quite accessible to folks who aren't necessarily in the biomanufacturing space. So a little bit later on, we'll be posting a link directly to this event so that you can register right away. And of course, you can always find more details about this event, the one that's taking place today, and all of the others on our cancersummit.ca website. And so on that note, just as a reminder, you can always find our previously recorded sessions on our website. So with respect to a few technical notes for today's session, we will be using the chat box and the Q&A functions. I'd ask that if you do have any questions, you use the Q&A box so that other participants can see the questions and can upvote any questions that they particularly like, and then I'll be able to moderate those questions if there are just simply too many to answer. With that being said, if you really prefer to ask your question verbally and orally, then just raise your hand and I'll unmute you. So there are those two options for asking questions. And then please use the chat box only if you have any sort of technical questions. And so with that being said, I'd like to kick us off by introducing the first two speakers. So the first one is Eva Villalba, and she is the Executive Director of the Quebec Cancer Coalition, a nonprofit patient advocacy group that's dedicated to improving Quebec healthcare system for people affected with cancer. And she'll be providing the patient perspective on today's topic. And then secondly is Abira Surendran, and she's a PhD student with Carolina Elkow, and she'll be presenting the plain language summary of the research that Carolina will subsequently be presenting. And then our final speaker is Dr. Carolina Elkow, and she is a scientist at the Ottawa Hospital Research Institute and an assistant professor with the Department of Biochemistry, Microbiology and Immunology at the University of Ottawa. So with that being said, I'd like to stop sharing my screen, and Eva, I welcome you to share yours. And thank you everyone for attending today.
Thank you. So today I'll be discussing the patient perspective on why this technology that we'll be hearing about today, the exosome-based cancer vaccines, have the potential to address the unmet needs of cancer patients. So I'm sure we're going to hear today, and you've heard already, about the promise of personalized medicine. And really the way that we think about it as in lay language is that it's a treatment tailored to the particular characteristics of an individual or their tumor, and therefore being more efficient and less toxic than what we normally have right now, which is the standard one-size-fits-all approach. If the first line of treatment doesn't work, then we'll try a second one, a third one, a fourth one. And during all that time, the patients are experiencing a lack of efficacy and also some really potentially serious side effects. And so personalized medicine really offers the promise that it will be able to give the right treatment at the right time for the right patients. And so we've seen lots of amazing breakthroughs lately in immunotherapy, in gene therapy, like CAR T therapies. And there's advantages and disadvantages to both from the patient perspective. So in terms of advantages, we know that it's a breakthrough. I mean, there's incredible efficacy, mostly in blood cancer so far, like in the CAR T therapies, they're all for blood cancers right now, so liquid tumors. They do significantly extend survival. There was just an article this week about this pediatric case that has completely been cured with CAR T therapy at Saint-Justin in Quebec. And then we're also using the patient's own immune cells to fight the cancer. And so it's really like personalized to the characteristics of the person and individualized. But the disadvantages are they're not yet applicable to solid tumors, which are far more frequent and more prevalent in the community. And so, we would want to address that significant frequency of cancer types in the population. There's really significant side effects and potential toxicity. So you have to like weigh the benefits and the disadvantages of doing that type of treatment. One of the main disadvantages is that it's extremely complex in terms of manufacturing, the logistics, the treatment process. It's like a long, complicated process that you can't just do anywhere. And so because of that, and because of the innovative nature of this treatment, it's extremely expensive, which means that it's not necessarily going to be available throughout Canada in every region. Our healthcare systems won't automatically be able to afford it. And then finally, because of all these reasons, it's really only available in very few centers across Canada. So what do patients want in general from their treatments? And how does this relate to both existing personalized medicine and the promise of Dr. Ilko's research? Well, first of all, they want efficacy, right? So can this get rid of my cancer? They want access in terms of availability. Is this something that I can find that is available and reimbursed in the hospital where I'm being treated? Proximity. Do I have to travel far for this treatment? Is it really close, or do I have to plan all of these logistical constraints for travel, for where do I stay, etc. And time. Can I actually afford to wait the delay that is given to me, or will my disease actually get worse? And then low or no toxicity. So when evaluating a treatment, it's always like, well, how will this affect me? Will I have some really serious side effects? Are they potentially deadly? Will I be able to tolerate them? And then finally, affordability. So can I, if I'm paying out of pocket, or my insurance or my hospital even afford this treatment? Or will I have to think of, for example, like we used to, have to go to the United States? And lately, we've been talking a lot about value-based healthcare, and precision medicine really falls well into value-based healthcare, because currently what we're doing in our existing healthcare systems is really looking at, what can the system afford? What does the system want? What is value for the system? But in a value-based healthcare perspective, value is really created and determined when you look at the outcomes that matter to patients, like the ones that I just described, divided by the total cost throughout the care journey. So currently we're only looking at how much a treatment costs, but not how much it costs to give the wrong treatment and then have the person rehospitalized. And so the reason that I bring up value-based healthcare is because any treatment that can provide better outcomes for patients at a lower cost really creates value for the patients and for the healthcare systems. And so that's something that we should absolutely keep in mind. So what are the current challenges? So even though there's like an amazing promise and potential for personalized medicine, there's a couple of challenges that we need to keep in mind. So the first one would be, it's going to come soon, there, regional and health inequities. So there's social determinants of health, there's regional disparities. When we're talking about a treatment that's only available in certain centers, usually urban centers, for example, or that are expensive, or then these regional and health inequities could be exacerbated. There's also barriers to market access. Because of the complexities of these new treatments, there's delays in the approval that prevent patients from having access. So sometimes patients hear about a promising treatment, but they don't have access to it right away. There's also the high cost of drug and treatments that impede speedy HTA approval and reimbursement. And so, when you have a treatment that costs $500,000, it will take a while to have access to that. Then there's toxicity and serious side effects, which I mentioned. So for example, in CAR-T, we know that there's potential cytokine release syndrome and neurotoxicity, which is very significant, and you have to plan to keep the patients close to the hospital in case they need ICU beds. And then there's the paradigm shift that is necessary to really change from a perspective of, just giving the standard of care to everyone and then seeing what happens, to from the beginning looking at what is specific, how can we use the patient's specific characteristics to better target the treatment. And so as our understanding of the mechanisms of action in cancer cells and immune response are changing, we have to also change the way that we make decisions. And now we're seeing, for example, something that's very promising from the patient point of view is having one medication that can treat several types of cancer, so pan-tumor treatments. And there's definitely an unmet need in solid tumors, like the main, breast cancer, lung cancer, prostate, colorectal, and all of the other solid tumors, because most of the innovations that are happening right now are in blood cancers or liquid tumors. And finally, sustainability. As exciting as precision treatments are, they're innovative and extremely costly for our public healthcare system. And it might not be sustainable to, offer only $500,000 treatments. And so how can we think of ways of making this more sustainable? Well, value-based healthcare and resilient healthcare systems must balance the cost and outcomes. So how can we get the best outcomes, the best health outcomes for patients for the lowest cost? And finally, I'll end by saying that this project, this team, based around the Dr. Ilko's project, is unique in the sense that, our coalition has been pushing to involve patients in clinical trials, and usually that's at phase 2, phase 3 clinical trials. But what's really unique about this team is that they're involving the patient perspective at the preclinical level and throughout, and we really believe that that's going to yield results that are much more patient-centered. So in conclusion, there is definitely an unmet need for personalized cancer treatments that are effective, affordable, accessible, with low toxicity, easy to manufacture, transport, store, and deliver, which would reduce costs, but also improve delays, and pan-tumor, so responding to a multitude of different cancer types. So thank you very much, and I'll pass it on to Abera, who will discuss more of the technicalities with you. Thank you.
All right, thank you, Eva, for the really great talk. So first, I'd like to start off by thanking Biotherapeutics for giving me the opportunity to be the plain language facilitator today. And I'll be giving an overview of the Engineered Biovesicle project in the Ilko and Bell Labs at the Ottawa Hospital Research Institute. So first and foremost, our lab is interested in oncolytic virus therapy. And what that is, they are genetically engineered or selected viruses that have the ability to selectively infect and kill cancer cells while leaving normal cells unharmed. And the way that's achieved is by taking advantage of molecular defects in cancer cells that prevent their ability to fight off the virus infection. So while both cells get infected, over time, the viral replication persists in cancer cells while the normal cells can fight off the infection. This ultimately leads to virus-mediated cell death. And all that inflammation in the tumor actually wakes up the immune system to provide anti-tumor immune responses. So oncolytic viruses are a really great platform because they act as replicating gene therapy vectors, meaning that they can be engineered to express all types of therapeutic cargo that we engineer into the virus. And historically, researchers have encoded all types of things into the virus. namely cytotoxic payloads. So you can imagine when a virus infects a cancer cell and you get replication of the virus, you also get expression of the cytotoxic payload that can help kill the cancer cells and provide a better anti-tumor response. Alternatively, researchers have looked at encoding cytokines, which are proteins that activate the immune system. And these cytokines can kind of help the host immune system fight off the tumor as well. And so the possibilities are really endless in terms of what we can encode into viruses with certain limitations. But our lab got interested in combining oncolytic virus therapy with the potential therapeutic effects of biovesicles. So what are these biovesicles? Well, it's kind of a broad term for small vesicles that may be referred to as microvesicles or exosomes that naturally bud off almost all cell types. And these small vesicles can carry all types of cargo, including proteins, nucleic acids, and so on. And really, they can act as a form of communication between cell types. And in the context of cancer, it's been shown that these small vesicles can be used by cells in the tumor microenvironment to communicate with each other. But interestingly, researchers have determined a way to take advantage of these small vesicles as a therapeutic delivery system. So you can think of it as a small package, and you can encode all kinds of things into this small package. But you need to tell the package where it needs to be sent. So often there are targeting ligands or targeting moieties that are expressed on the surface of these vesicles to tell the vesicles where to go. A lot of the time, this is intended for cancer cells, and so you may target a specific tumor antigen. But other times, you can actually target immune cells to help their anti-tumor function, kind of activate them or engage them in the anti-tumor response. So in addition to providing this address or targeting ligand, you can also encode all kinds of therapeutic cargo. This can include drugs that can help fight the tumor, but again, it can also include biomolecules such as proteins, DNA, RNA, and so on. As a therapeutic delivery device, currently, there are two kind of ways that it's administered to the patient. So the exogenous method starts with growing cells in a flask and harvesting the vesicles to be transferred into the patient. But to truly give a personalized approach, the autologous approach involves taking these vesicles from the patient, manipulating them in a way to give it a therapeutic advantage, and then re-administering into the patient. And this can often be expensive, resource-intensive, and time-consuming. So our lab sought to take advantage of the fact that these viruses can hijack the host cell exosomal pathway to export their own products. And so we hypothesized that it would be possible to exploit this naturally occurring event for therapeutic gain. So in other words, as I mentioned to you previously, these viruses can be genetically altered to encode specific therapeutic cargo. And so in our platform, we try to encode the pieces that you would need to generate these biovesicles that naturally are induced by virus infection to contain specific components, such as the address or targeting moiety and the therapeutic cargo of choice. So you can kind of consider this analogy, where you have the first biotherapeutic, which is the oncolytic virus, which infects the cancer cells and replicates in the cancer cells. But as it does so, it's expressing the second biotherapeutic that we've now encoded into the virus. And so you get this in situ, or at the site of the tumor, the production of these therapeutic biovesicles, which actually acts as almost a personalized therapy within the tumor itself. But an interesting advantage of these biovesicles also is that you can get local delivery within the tumor microenvironment. So you can imagine, once you have the delivery of these targeted biovesicles, you can even reach cells that may not have been infected by the oncolytic virus, for instance. And in the context of engaging immune cells to help fight the cancer, you can encode immune cell agonists as the therapeutic cargo, which could help the tumor eradication. So really our approach is taking the best of both worlds, using a virus that can selectively infect and kill cancer cells, and using these biovesicles, which can enhance local delivery of therapeutic cargo in the tumor microenvironment. And together they can both really contribute to a more robust anti-tumor response. And with that, I think I'll pass on the torch to Carolina.
Thanks very much, Abera. That was wonderful. We do have one question in the Q&A, and it's for Eva. So I'm not exactly sure when this question came in. So Eva, maybe you can nuance what the person is asking about. But the question is, what's the difference as far as cost is concerned? So at the beginning, it might have been with respect to car products, but maybe you can.
Yeah. Well, I mean, you know, we never know exactly how much these drugs cost because they have special conditions and negotiated prices. However, we know that the estimate is like for CAR T therapies, it costs approximately $500,000, $500,000, which is enormous. But it's also because, like Abera showed, so it's an autologous type of treatment where you have to go through the process of extracting the patient's immune cells. Then you have to ship them to the United States. It takes a couple of weeks. They're re-engineered in the lab in the United States, and then they're shipped back. And so that whole process, and there's a whole like, you know, cold chain that has to be maintained, and the storage is complicated, and the logistics. And so that's very expensive. And so if there was a possibility to have a treatment like Carolina Ilka is going to describe that can be given with the patient's cells directly in you know, at the hospital where the patient is, without need to like send it off and ship it, and you don't have to go through that whole process, it would naturally be much less expensive and so more affordable for our healthcare systems. I hope that answered the question.
Thank you. And if not, if I invite that person who asked it, we have time, a few more minutes. Oh, perfect. Yeah, that's great. So I think it answered the question. And we do have a few more minutes before Carolina's talk. And there is another question that came in for Abera. And I think Carolina will probably speak to this too, but nonetheless, maybe wet everyone's whistle. So the next question is this technology targeted towards targeted towards solid tumors, or is it restricted to liquid tumors? And I think that's an exciting question and answer.
So far in our lab, we've been actually focused on solid tumors, but I don't see why this technology couldn't work in liquid tumors. But again, Carolina will probably elaborate on that.
Perfect. Okay. Yeah, so I think those are all the questions that have come in so far. Of course, I do welcome more, and I think we all welcome more questions to come in as the speakers are chatting, and I'll keep monitoring the Q&A. But I think at this point, we're a few minutes early, but why don't we jump right into Carolina's presentation and take from there, and then we can have more time for questions perhaps at the end of the session. So welcome, Carolina.
Thank you. I think, Abera, maybe you need to stop sharing your presentation. There you go. Screen. Okay. Is this okay?
Yeah, looks great.
Excellent. Thank you. Okay. Hi, everyone. I'm super excited to be sharing with all of you our platform and our current research that in part is supported by biokinetics. So we are very grateful for that, and also excited to share and what we are thinking, how to move this forward, and welcome to new collaborations as well as feedback on what we are thinking. So as Abera and Eva alluded earlier, we are working on these biovesicles that we are modifying them to use them for cancer immunotherapy. And I will be walking through a little of these ideas for my presentation. So before I get into that, I have to really show you who is doing the work, because it's not me. So first of all, this is a collaborative project. So we are working very close together with Dr. Brian Lichty at McMaster University, as well as with Dr. John Bell. And the people that are here are the ones that indeed, in some shape or form, have been involved in the data that I will share with you today. But the team is larger than this. We are also very grateful for all the trainees that are helping in these projects and are so motivated to work in exosomes. So let's get started. So before we jump into biovesicles or exosomes and cancer-killing viruses, we really need to acknowledge that there is a big array indeed of biotherapeutics. And biotherapeutics for cancer treatment are mostly those that are specific for cancer, to kill cancer in some shape or form, but also are therapeutics that are really good at stimulating the patient's own immune system to fight the cancer. As I mentioned, there are multiple types, including monoclonal antibodies, and we know how successful for certain solid tumors checkpoint inhibitors are, and these are very powerful biotherapeutics. Tumor vaccines also. So there are multiple types of cancer cell or immune cell-based cancer vaccines available. Of course, the CAR T cells or other cell-based therapies that in certain cases are very successful, like Eva was mentioning, for liquid tumors and so forth. Also, there are bacterial therapies. So there are multiple examples where bacteria are being used to stimulate the immune system and in that way fight the tumor. But the ones that today I want to really touch and describe to you is the use of biovesicles. And biovesicles is a very generic term that includes perhaps the most famous exosomes, which are the ones that are being used as therapeutics. But to be honest, the generalized term is biovesicles, which encompass all the extracellular vesicles that are produced by the cell. And the beauty of these biovesicles is that they are very easy to be modified. So they can be reformed or reshaped to carry different drugs or therapeutic payloads, but also they can be decorated to be targeted or delivered to certain cell types in the tumor microenvironment, including cancer cells, as well as immune cells or other components of the tumor ecosystem. And the other example of biotherapeutics that I will also be describing today in more detail are viral therapies, including cancer-killing viruses. So let's get started with biovesicles and specifically with exosomes. Before we get into why these are powerful therapeutics and very promising therapeutics for multiple diseases, not only for cancer, indeed, biovesicles are being explored to treat Alzheimer's, for wound healing, for multiple lung and respiratory problems. So the promise of biovesicles as therapeutics is, of course, they are for cancer, but also because they are so versatile, they can be carriers of whatever therapeutic indeed we want. And the actual power of these biovesicles indeed starts by the fact that all our cells in our body communicate all the time with the language that indeed is biovesicles. So cells communicate among each other, cells that are the same type, but also heterotypic interactions, so cells of different nature also communicate via these extracellular vesicles, including exosomes, microvesicles, and so forth. And many years ago, we used to think that these vesicles indeed are like garbage bags where whatever the cell didn't want will be into these vesicles for disposal. And now more and more, indeed, we learn that whatever it goes into these vesicles is it has a function. And indeed, not all the vesicles are the same. There are, as illustrated in this cartoon, depending on the cell type, the biovesicles or the exosomes will look different and will contain different cargo. And this is very important also because allows the how versatile these vesicles are, so they can load different cargo depending on the situation. So a little more about these biovesicles, so we really understand what they contain. So this is a schematic representation indeed of a generic extracellular vesicle. This doesn't mean that all the vesicles contain all these components, but this is mostly what they tend to have. So they have proteins, and they have proteins in the cytosolic part of the vesicle. And those are proteins that, as I was mentioning earlier, are purposely loaded into these vesicles for a function. But also there are proteins on the surface of these vesicles. So these vesicles are decorated by proteins that will give the information of these vesicles where to go, where to attach, with which cells to interact. So there are membrane proteins associated with these vesicles. But in addition to proteins, most of the cargo indeed of the naturally occurring vesicles is nucleic acids, and specifically microRNAs. So those small RNA sequences that have the power to regulate protein expression. But also other RNA species are found in these biovesicles. So this is how they look in natural situation in our body, and they are all the time circulating. They, as I mentioned, they have a specific information. They are even being used currently for liquid biopsy. So they have the promise behind the biovesicles is even beyond the therapeutic aspect. There are a lot of groups that are indeed doing liquid biopsies and are mapping how these vesicles look like, and using that to early detection of cancer, for example. So the promise is beyond therapeutics. But today we will focus on the therapeutic angle of these vesicles. So I show you the naturally occurring vesicles. So those are circulating in our body. This is how they look when they are modified. So as I alluded earlier, these vesicles are now powerful delivery system for drugs and for therapeutic payloads. These are some of the examples that indeed illustrate what drugs in the upper part, so multiple chemotherapeutics indeed are being loaded in exosomes, and those exosomes are carrying now these toxic drugs to the tumor instead of being distributed generally in the entire body. So there is this specific delivery of chemotherapeutics using these biovesicles. But also there are a lot of microRNA and different nucleic acid therapeutics that are being delivered using these biovesicles. And I will give you an example that is of clinical trials indeed that are happening, for example, in MD Anderson as we speak, to target KRAS mutated tumors with biovesicles loaded with a microRNA that is specifically target mutated RAS. So these are just key examples of how these therapeutics are being used. And of course, in addition to the delivery of RNA, they can also deliver therapeutic proteins, including cytotoxic proteins, including proteins that can stimulate the immune system and so forth. And I will show you a couple of examples during the presentation. So that is the biovesicle as a therapeutic. And this is just a short list of some of the clinical trials that indeed are currently taking place or about to start that involved exosomes as therapeutics. And I highlighted in the red boxes the ones that involved the use of exosomes as therapeutics for different cancers. There are, if we have to generalize, they tend to be used as to stimulate the immune system. So the first clinical trials using exosomes for cancer treatment indeed have been those that were exosomes, autologous exosomes, loaded with a specific cancer or tumor antigens, and they were then injected into patients to re-stimulate the immune system. So they acted almost as a vaccine. But as I mentioned a little earlier, there are some new, indeed, approaches like the one in the MD Anderson that indeed, in this particular case, is loading the exosomes with different microRNAs targeting KRAS. And that is for metastatic pancreatic cancer. And that is a very exciting piece of work from the Kaluri lab that I recommend everyone to read. It's a very comprehensive study. And even the manufacturing part of those extracellular vesicles is well illustrated by the group. So it probably is one of the most advanced groups in terms of developing exosomes for cancer treatment. But as you could see, there are not many. Exosomes are, at least in the cancer field, is a new platform, and we have a lot to learn, not only of how to make the best use of these exosomes, but also how to properly manufacture them. And I will touch a little more in the next slides about that. Indeed, it's in here. So I think the key thing with these biovesicles is they are super powerful, and the power is that they are invisible to the immune system. So if they are made, specifically if they are made from patient-derived cells, and those could be tumor cells, it could be monocytes, for example, the dendritic cells, as well as MSCs. In those particular cases, the exosomes tend to be invisible. So we can give the exosomes as many times as we want to the patient, loaded with whatever therapeutic cargo we want, that the immune system will not react. Therefore, we can do multiple dosing. And that is super powerful. The other advantage, I will say, of biovesicles is that they can be lyophilized, so they can be stored at room temperature, and they last for a while, so they are easy to manage. But the problem is still that they had to be autologous, so they had to come from the patient's own cells, and those could be tumor cells or monocytes or different cell types, but still require a lot of laborious work, and is very personalized. And it's not only that, is once these cells are obtained from the patient, then the extracellular vesicles had to be, or these biovesicles had to be purified, and then they had to be loaded, and then reinfused into the patient. So in addition to all these complicated and very laborious and time-consuming process of obtaining the vesicles, the other key problem of making these ex vivo and then reinfusing into the patients is how to properly make them, how to properly load them. And in the next slide, I think I summarize in this cartoon some of the key ways of how this can be done. So how we can encapsulate therapeutics in these biovesicles, and also how we can decorate the vesicle to be targeted to the specific cells. So in the majority of the cases, indeed, it happened by modifying or genetically modifying the patient's cells ex vivo. So obtaining the cells and then using, for example, lentiviruses or adenoviruses to modify the cell and make, in that case, the cell to express the therapeutic cargo or the signal that will be displayed in the vesicles, and then purify the vesicles from there. In other cases, instead of doing that, other methods are used as the ones illustrated in the bottom part of the cartoon. So electroporation can be used, different free and so cycles to enhance the uptake of certain, for example, drugs into these cells and so forth. So there are different methods depending on what is the therapeutic cargo and how these vesicles had to be modified. But I think the point that I really want to make is that this is very laborious, and this is time consuming, and this costs a lot of money. And in certain cases, indeed, some of these methods are not even very efficient. So with this in mind, we start thinking and really going over the literature to see if there was a possibility to combine our cancer-killing viruses with this biovesicle approach. And I think, as Vera mentioned during her late audience talk, there is a connection, and there is a connection between viruses and biovesicles. And the first one is as simple as cell communicating in a language that looks like viruses. So when we take a look at these exosomes, at these biovesicles, they are very, very similar to viruses, especially envelope viruses. Not only that, is like these biovesicles almost use the same machinery, or the viruses use the same machinery that these biovesicles use to be produced from a cell. So there is a lot of similarities between a virus and a biovesicle. But not only that, is that when we dig a little in how viruses affect the host cell, one of the things that all the viruses, or most of the virus, will do is hijack, indeed, the exosomal pathway. And they do that to export their own proteins or their own information to modify the infection site. So, and I think this is shown in this cartoon that clearly shows that first of all, a virus and these biovesicles use similar pathways within the cell. And in addition to that, here is shown that multiple viruses will express components that end up into these vesicles. And this, of course, is for the advantage of the virus. But on the other hand, these gave us the vision to hijack the pathway or trick the virus to, instead of loading components that are good for the virus, how come we thought to modify these and indeed load therapeutic components that are expressed from the virus, but now they can end up in these vesicles. So I will explain this a little better in the next couple of slides to clarify. But this is the key thing that makes us thinking that this was possible. So you know that we work with viruses, and specifically we work with cancer-killing viruses or oncolytic viruses. I'm sure most of you are familiar with this, but just to remind you, these are viruses that are selected from nature, or we engineer it to preferentially infect, grow, and kill cancer cells. But in addition to that, more and more we are learning that these viruses can do more than killing cancer cells. One of the things that these viruses do very, very well is stimulate the immune system. And this is just summarized in this cartoon, where it's shown that these viruses, yes, when they are administered, they home to the tumor because they have affinity for growing into this tumor and growing there and replicating. But also they are able to kill cancer cells and release tumor antigens and attract immune cells to the tumor site. And in that case, boost the immune response, and eventually they act as cancer vaccines in situ. So they are able to spread tumor antigens, as well as call in and educate the immune cells to act against the tumor. So they are very powerful at doing both things. But in addition to this, indeed, the other key aspect of the viruses that we work, these cancer-killing viruses, is that they are also vectors. And they are, what I refer by that is that these viruses, in addition to infect the tumor and stimulate the immune system, they can also carry genetic information to express a payload, to express a transgene that could be therapeutic. And in that regard, there are multiple examples where we know that these vectors could express therapeutic microRNAs, they can express prodrug converting enzymes, they can express different cytokines, different monoclonal antibodies. So they are also vectors, and they can carry that information in the tumor, and they can express it in the tumor. But the other thing that I mentioned to you earlier is that in addition to that, now we know that these viruses can also hijack the exosomal pathway. And they do that in nature. It's not like we created this. Nature has done it for us. And what we decided to do now is to hijack that. So to trick the virus to load into the exosomal pathway, not its own products, but now it has to load these therapeutic cargoes that are indeed expressed from the virus, because the virus is the vector. So this is the power of oncolytic virus. It's not gene therapy, but in this, it's almost like a gene therapy add-on to the already existing features of the virus, which is killing cancer cells and stimulating the immune system. So this is the overview. So if there is one slide that I hope to really make a point today, is this. Indeed, I will use this slide twice because of this is what we are doing. So the overall picture is we work with cancer-killing viruses. This is one product. And these viruses are known and are specialized at infecting cancer cells and killing cancer cells, but also are vectors. And are vectors that can carry genetic information to modify the biovesicles produced by the infected cell in a way that they can carry a therapeutic cargo, but also they can display or be decorated by with information to target these vesicles to specific cells in the tumor microenvironment. Those could be other cancer cells. For example, if we want to express a cytotoxic payload, it could be components of the tumor stroma, like cancer-associated fibroblasts in fibrotic tumors, or it could be immune cells in the case that we want to stimulate the immune system or educate the immune system to fight that tumor. And I will show you examples of both during my next slides. But this is the overview platform. So the virus, in addition to kill cancer and stimulate the immune system, can indeed hijack the exosomal pathway, and it can indeed in situ fabricate or make extracellular vesicles or biovesicles that are loaded with a therapeutic cargo, and that they are targeted to specific cells in the tumor microenvironment. So why this matters and why this is important? In addition to the power of what we think is boosting the activity of the oncolytic virus, as well as the exosomal therapeutics, I think it has also a cost-benefit angle. More and more we are learning that the best way to treat cancer is combination therapies. And we know that it's not only combination therapies, but also is immunotherapies. And in multiple cases, personalized therapies as the CAR T cells, as the exosome-based platforms that are being tested in clinical trials currently. Those are personalized. And they have great promise and it's fantastic, but it's very expensive. And so what we thought is that can we arm oncolytic viruses in a way to produce one product that will have the function of two? So in other words, the power of oncolytic viruses is that, as I mentioned earlier, not only they go to the tumor, they kill cancer cells, they stimulate the immune system, but also they can produce a second therapeutic within the tumor. And that second therapeutic, that in the case that today I will be describing, is the exosome that is loaded with a therapeutic cargo to expand the cancer killing activity of in this case, the virus. So in other words, is one product, is not personalized, but when it goes to the tumor, will indeed modify the exosomes, and that is personalized because the tumor now is the factory of the second therapy, which are these exosomes. Okay, so this is the promise, and now I will give you some examples of the work that we are doing to hopefully soonish get to the clinic and be able to have a product that is innovated in the way that is affordable, is personalized without being personalized, and hopefully it will work very well in solid tumors. So these are our biovesicles. So before we get into we have the oncolytic viruses, we know these platforms very well, but then we need to come up with how we will indeed modify in situ in the tumor these vesicles. And we have different flavors. We came up with different ideas. We tend to call these biovesicle targeting devices. So mostly are chimeric things that we have created. So this is synthetic biology that allowed us to, at the moment, have different flavors or different platforms of these biovesicles. And today I will be giving you examples of 3 of them. One is our Exosome platform. The other is our Exosome activating platform. And the last one is what we call virus-like particles that instead of carrying proteins, they carry genetic material to, in that case, carry the mRNA and be able to produce protein in the recipient cells. So the first example is these exosomes. And in order to do this, we go back to our platform and we adapt it for expression of different microRNA, therapeutic microRNAs. In this case, we have oncolytic viruses that will express a therapeutic microRNA. And that therapeutic microRNA is because we build it in a cassette that is enriched in exosomes. That microRNA is particularly enriched and shuttled to these vesicles that then will be uptaken by different cancer cells in the tumor microenvironment and play the role, the therapeutic role that we expect. So this is the overview of the platform. But to get to really polish this platform and improve this platform, we couldn't pick any microRNA. We have to be any cassette. We have to come out with a cassette that will be enriched and really optimized to be delivered into exosomes. So towards doing that, the first thing that we did is we used different oncolytic virus platforms, and we use indeed 3 very different oncolytic virus platforms, all being tested in the clinic. And what we ask is, if we infect cancer cells and multiple cancer cells, we use breast, we use pancreatic, we use kidney, renal cell carcinoma, so multiple cancer cell lines. And then we collect the extracellular vesicles, what microRNAs are in those vesicles. And the idea behind that, it was to identify what microRNAs are enriching those exosomes to then genetically modify them to become therapeutic. So in other words, we were looking for a cassette to do this job. And there are many microRNAs being expressed, so I don't have the names here, but this is the type of data that we generated. So there are microRNAs that are exquisitely enriching exosomes. They are almost not present in the cells upon infection. They are all gone into the extracellular vesicles. And on the contrary, there are others that are retained in the cell. So for us, the ones that we really care are the ones that are sent off from the infected cell, because that is the cassette or the microRNA that we want to use as a cassette and as a therapeutic platform to move forward. So as I mentioned, we did this for 3 different oncolytic viruses, and then we identify multiple cassettes, but we are focusing on 8 for various reasons. One is because they are representative in multiple tumor types, and when we infect multiple cancer cell lines, as well as they are, it doesn't matter which oncolytic virus we use, indeed the overexpression in the exosomes, it happens in this case, in the 3 platforms. So with that in mind, we selected one cassette, and then we modified that cassette, this particular microRNA, to express a very tiny piece, the short herping RNA, to target PD-L1. And once we did that, we expressed this into an oncolytic virus, in this case, BSV Delta 51. And as shown in panel A, what we were able to demonstrate is that although PD-L1 is upregulated upon infection of cancer cells, and this is something that happened almost with all the oncolytic viruses, is part of the interferon response that is stimulated by the virus. But when we express an oncolytic virus that targets PD-L1, PD-L1 is gone. Really showing that this short herping is functional and is doing what we are supposed to do. Now, at this point, this doesn't mean that it's going into biovesicles. So then we have to do some experiments that indeed will address that. And the way that we did this is by using viruses that, even though can infect the cancer cell, they cannot produce new virions. So they are replicating but not spreading viruses. And we did this just to be able to separate the extracellular vesicles from the infectious particles and really demonstrate the power of the technology that we are developing. So in this case, we infected with these non-spreading viruses a transwell system. So we infected the upper part, the cells that are attached to the transwell, with this virus that express or not the microRNA with the short herping PD-L1. And then we transferred this transwell to cells that were not infected. And the idea behind that was that although these cells are infected, new virions cannot be produced because we use this specific not spreading virus, but the extracellular vesicles, so the biovesicles produced by these cells, will go through the transwell and eventually will reach the monolayer on the bottom. And this was a way for us to really show if the short herping was or not enriching these extracellular vesicles. And as shown in the Western blot, using these delta G viruses or non-replicating viruses, we were able to demonstrate not only that we have a downregulation of the short hairpin in the donor cells, so the donor cells are the one in the upper part, but also we observed a downregulation when the cells are exposed to these biovesicles. And of course, in the virus that is expressed in the short hairpin PD-L1, but that does not happen in the control. So this was the first type of experiments that we did in order to demonstrate that indeed we can modify viruses to express exomeres. And those exomeres can have a function in cells that are not infecting. So really showing that bystander effect. So this platform works in vitro and it shows here the power in vitro, but also we have therapeutic advantage when we use this platform, this OV platform expressing the short hairpin PD-L1 in exosomes. As shown in here, this is the survival curve of B16 tumors injected with control virus or with a virus expressing the short hairpin PD-L1 built in an exomere cassette. Some of the animals indeed survived. We rechallenged those animals as shown in B, and indeed none of the animals that were cured through the treatment, once we rechallenged them with tumor, after 100 days of survival, none of them developed new tumors, indicating that indeed the effect of the therapy was mostly by inducing anti-tumor responses that build long-lasting anti-tumor protection. So this is our Exosome platform, and this is just an example of the type of work that we are doing in this angle. The other platform that I really want to share with you, and this is the project that we are working together with Dr. Brian Lichty and McMaster and Dr. John Bell, and we are supported by Biotherapeutics. So this is a very exciting project, and I hope you understand why we are so excited and so motivated to move this forward. So this is a platform that instead of transporting exomeres, we are transporting, in this case, a functional enzyme. And in this case, is a functional enzyme that will activate the STING pathway in immune cells. And we call this platform exocyclase, and you will understand a little later why we call it in that way. So the way that this platform is built is, as you will see, is a platform that is, the purpose is to stimulate the STING pathway. Now, why we want to stimulate the STING pathway? So the STING pathway, as probably all of you know, is extremely important. It's extremely important for fighting cancer. Indeed, the basis of this pathway is an immune sensing pathway that can recognize and act once DNA is found in the cytoplasm, and that could happen by, for example, infections. It's also infections, viral or bacterial infections, but also it can recognize self-DNA. And the important part is that this pathway, as shown in the next slide, indeed is many times in many tumor types, for example, ovarian cancers, colorectal, melanoma, and so forth, this pathway is somehow impaired, so it's not functional. And this is indeed a strategy or a mechanism that tumors have to avoid self-DNA mediated immune activation. So in other words, it's just to silence the immune system. Because of this, there are many groups and approaches to re-stimulate or stimulate the STING pathway for cancer treatment. So this is another way of stimulating the immune system and another platform of cancer immunotherapy. And those are called STING agonists. So these are indeed molecules small molecules that can reactivate the STING pathway in the tumor microenvironment, and in that case, boost tumor responses. Indeed, there is data that show that direct activation of the STING pathway in the tumor microenvironment by direct injection can indeed boost long-lasting immunity. And there are multiple clinical trials of drugs that are being tested to do this job. So indeed, it's a very hot topic currently in immunotherapy, the use of different approaches to stimulate the STING pathway, mostly by using these agonist molecules. Now, there are barriers to success, and even though stimulating the STING pathway using these molecules is super exciting, and indeed in preclinical work is the data is very convincing and strong, there are some complications that make these agonists to advance to clinical trials. One is that indeed they have a lot of adverse effects. So in other words, they are very toxic. And because they are so toxic, they cannot be given systemically. So in other words, they have to be done by intratumoral injection just to try to limit the toxicity associated with these agonists. And mostly the toxicity comes for the fact that when they are given, these STING agonists are not tailored to cancer cells. So in other words, they can activate this pathway, not only in the tumor tissue, but also in normal cells. And that is what is indeed the main reason of the high toxicity observed. And so far, even though the promise is huge, the efficacy and the activity of these drugs so far is not as powerful as we expected. Therefore, there are a lot of groups that are considering using these STING agonists in combination with other immunotherapeutic approaches or other therapies. So what we thought is, can we combine some molecules that will activate the STING pathway with oncolytic viruses? And before doing that, and this is the work done by the Brian Lichty lab, is we need to find the best molecule to activate the STING pathway. And I think something that is now very evident is that indeed the STING pathway has evolved from bacteria. And bacteria already had some molecules or some enzymes that were able indeed to play the role of a C gas, which is a key core component of the STING pathway. And these enzymes, which are bacterial, C gas-like enzymes, are very similar to the C gas present in human cells and very often lost in cancer, in the cancer microenvironment. So what Brian Lichty and his team did is indeed screen an array of different bacterial cyclases, which are these C gas-like enzymes, and ask the question, can we identify bacterial C gas-like enzymes that will activate the interferon pathway or STING in cancer cells? And the bottom line and the answer is yes, we could. And not all of the enzymes tested were able to do this, but indeed 3 of them were very good and very efficient at stimulating the STING pathway. So this was super exciting. And with that information in mind, what we decided to do is, can we now express those STING activating cyclases from an oncolytic virus? So in this case, it's a vaccinia virus. And we selected 2 of them, and we expressed it from our oncolytic virus platforms. And we were able to demonstrate that indeed viruses that express the cyclase is identified by the Lichty lab, indeed were very good at stimulating the STIM pathway without compromising the activity of the virus, so the growth of the virus and the infectivity of the virus. This is shown in panel B, in panel A, and as well as in panel B, where we have a strong inactivation of the STIM pathway that's shown by the phosphorylation of STIM, as well as all the downstream molecules that act in that pathway. But what is very important is that in cells that do not express C gas, which is this is what normally happens in cancer. So many cancers will lose C gas, as I mentioned earlier, as a way of immune escape. These viruses that express these bacterial cyclases, but not the control virus, are able to stimulate STIM very efficiently. And I think this is the power of this technology, that even in tumors that do not have C gas and will not have activation of this pathway, now with this virus, we can activate it. Not only that, this virus also is more efficacious in vivo, as shown here in a very challenging tumor, colorectal tumor. This is MC38. We have better therapeutic responses with the virus when it's loaded with this cyclase. Now, one of the things that I mentioned earlier is that activating STIM is clearly very powerful, and is something that many groups are trying to do. But also earlier, when I was talking about the agonist, I alluded that that could be toxic because it's not good to stimulate this pathway or in healthy cells. So thinking in our Biovesicle approach, we decided to take one step further. And this is, can we make these cyclases to be specifically loaded into biovesicles produced by the infected cell? So in other words, is instead of expressing the plain cyclase, can we express the cyclase linked to our biovesicle devices to now, instead of sending free cyclase to activate STIM in the tumor microenvironment with the potentials of off-target effect, can we now hide it into these vesicles? And not only hide it into these vesicles, but also can we send it to the specific cells in the tumor microenvironment that we want to activate? So in other words, can we send these to dendritic cells and in that case stimulate the anti-tumor response? And this is what we did. We make chimeric devices that, in addition to have our STIM activating molecule, also have a signal that will target this into extracellular vesicles, and also has a single chain variable fragment that in this particular case recognize the DEC205 receptor in the dendritic cells, and in that case these vesicles are specifically targeted to these dendritic cells. So in other words, we have the address for the vesicle and we have the cargo. And the function of these vesicles will be activate dendritic cells to stimulate antigen presentation and T-cell responses and immunity in this tumor. So the answer is yes, we can do that, and is what we are currently working on it. But I just want to show you, because this is very, in my opinion, very exciting data that we really want to move forward. So as I mentioned earlier, the biovesicle, the exocyclase that now is being expressed from the virus, it contains these devices that has the cyclase, which will activate the STIM, has the exo-targeting moiety, as well as, in this particular case, a single chain variable fragment antibody that will drag these vesicles to dendritic cells. So the first Western blot shown here is just to show you that this is doable. that everything is being expressed properly, is being expressed from the construct per se as a validation, is expressed from the virus very properly. And then when we purify the vesicles from the virus, just to really show that this construct is loaded, we observed the exocyclase also in purified exosomes. After doing that, we quantify the exosomes. So we use technology, ZetaView, that allowed us to quantify how many exosomes we have. And in this case, we use around 800 or around 2,500 exosomes per dendritic cell. We purify, and this work was done by Juan in the Lichty lab. We purify the exosomes, and also we purify dendritic cells. And what we did is we exposed the dendritic cells to these exosomes, and we did a dose response. And we have some multiple controls, which include a dead cyclase, which shouldn't activate STING. Instead of a cyclase, we have the green fluorescent protein, or we have a construct that is not targeted. And then we have our exocyclase construct. And the key thing here is that in a dose fashion, we have activation of the STING pathway only when the exocyclase is expressed in this, or is contained in these exosomes. And more importantly, when we do the same exercise, but instead of using dendritic cells, we use fibroblasts, which are known for not having DEC205 on the surface. So this will be an example of cells that we don't want to harm, that we don't want to activate the STING pathway. Even though we add the same amount than here of exosomes, so this is the same prep, there is no activation of the STING pathway by this exocyclase. And then we use two different controls, like transfection of DNA and so forth, that will stimulate the STING pathway. So the STING pathway can be stimulated in these cells, but our vesicle, because it's not being uptaken by these cells, there is no toxicity. So this is the power, and is where we are going. The last set of slides is, I have two more slides and I'm done, is just to show you that in addition to these biovesicles, we can also use what we call VLPs. So these are virus-like particles. They are very similar to these biovesicles, but now we are loading messenger RNA. And this is indeed work that Nicholas and Tahar are really moving forward in the lab, and it's another clear example of how this platform can be used. And in this case, the idea is that the virus will express all the components to form these biovesicles that are VLPs, as well as the information to have this self-amplifying RNA expressed and loaded into these vesicles. And as a proof of concept, Nicholas had made one construct that is expressed from the virus, in this case, again, a vaccinia virus that is green. And then he put all the information to form these biovesicles containing the self-amplifying RNA for a red protein. So in other words, we have infected cells, which are shown in green or yellow. And then the biovesicles will contain the genetic information to express the red fluorescent protein in cells that were not infected. And this is a clear example of this bystander effect of the technology that goes beyond the infected point. This is true in a monolayer of cells. In this case, is the transfer of the vesicles without virus, and showing that indeed are the vesicles that are inducing the production of this protein in cells that are not infected. And in addition to do this in monolayers, we also are working with using spheroid models. And I really like this because it's the example of this bystander effect. So in this spheroid or 3D model, you could appreciate that when the spheroid is infected with this vaccinia virus, oncolytic virus that express the yellow fluorescent protein, or in this case looks green, but the infection is all around the core of this spheroid. But when we use the VLP virus, so the virus that in addition to express the green, is in addition of the green, we can observe that the VLPs are also present in the core of this spheroid, really showing that the vesicles go beyond the infected cells. With this, this is what I wanted to share with you today. So these are some examples of the combination of oncolytic viruses and biovesicles for delivery of therapeutic cargo. As I mentioned earlier, this is indeed the slide that summarizes all the work that we are doing. So we are using viruses to reprogram these in situ, in the tumor microenvironment, these vesicles with therapeutic information, but also displaying or being decorated by single chain variable fragment antibodies or other molecules that will drag these vesicles to target cells, and it's specific in a very specific way that will reduce toxicity and also enhance therapy. So with that, I would like to thank you, all of you, for being there, for listening, and all the collaborators, especially Dr. John Bell and Brian Lichty, for being core members of this project. The funding will not be possible. Thank you to Biokinetics, who is supporting our Exocyclase project, and also to Avera and Eva for teaming up with me and explaining the patient perspective as well as the lay language of this project. And I will be happy to take questions. Thank you.
Wonderful. Thank you very much, Carolina, for a really good synthesis of a lot of data and a lot of different strategies. So we do have a couple of questions that came in, and I do invite others to either ask a question in the Q&A, or if you prefer to ask it orally, you can always raise the hand and I'll unmute you. But the questions that came in, the first one is more general, I think, but it is, liposomes have been used in drug delivery for years. What are the main differences and advantages of exosomes versus liposomes?
So it's a great question, and I don't think in multiple cases have been compared head to head with the same in the same situation. So maybe it's unfair what I will say, but I think the advantage of the exosomes is that they are naturally occurring, and they have that everything is balanced, and is balanced in a way that is so they happen in the body. So when they are, even though they are modified in one way to be therapeutic, but they are still not recognized by the immune system. They don't have toxicity. They last a long time in circulation. So I think those are the advantages compared to the synthetic nanoparticles like liposomes.
And the next question is, what is the implication of cancer reoccurrence? Will the immune system have adapted to the first exposure so that additional treatment for cancer recurrence will not be as effective as in the first time?
Yeah, so I don't know, maybe the person can clarify if it's related to the biovesicles or the viruses. So, but maybe I will answer both, and hopefully is sufficient. So the problem with oncolytic viruses and any viral platforms is that it's very hard to multi-dosing, because eventually the virus is like vaccines, right? Like our immune system will recognize these foreign, in this case, viruses and mount immune responses, and then the virus, if we want to reinfuse or re-administer the virus. Unfortunately, the immune system will neutralize the virus, so it's not possible. Now, there are multiple strategies that are being developed to change the flavor or the look of the virus, so to trick the immune system and to re-multidose. So it is possible. Now, with the biovesicles, I think that is the power that they have. It's like they are inert. So I have heard talks of people multidosing 100 days with exosomes and not alerting the immune system. And I think that is the power, and why everyone is so excited is because there is something that happened in our bodies. So there is no way to fight for it because it's natural. And as long as the genetic modifications don't change the vesicle that much, multidosing is totally feasible.
Great. And we have a couple of questions coming in that will be asked orally. So Priya, I turn to you.
Hi, thank you so much. The reason I raised my hand rather than using the chat is because I wanted to kind of introduce myself. My name is Priya. I also work on exosomes and cancer, and I'm specifically looking at macrophages that can actually, they're specialized for the uptake of these particles. And so they clear it out of the tumor microenvironment, for example. So I was very excited to see your DC data where we expect in macrophages actually for the exosomes to just go into the endosome and just be degraded and never meet any C-gas sting in the cytoplasm. So that in DCs was really nice to see. But my question though is a little bit more kind of technical. This is regarding your PD-L1 system. Like, do you have any controls in your system to help you understand the differences between the effect of the virus versus the effect of the exosome? For example, do you have a virus that has SH-PD-L1 not in the exo-MIR construct? Are there, like, just because the virus is carrying SH-PD-L1, you might see that effect, right? So are there any other ways in which you can tease out, like, what's the effect of the virus versus what's the effect of the exosomes?
Yeah, so totally. So we have multiple, so we have NTCs and we have multiple other flavors that we have compare and contrast. But so we have controlled that by using that type of assays. But I think the most important piece of data that we have is in part what we showed with the transwell system, where there is no transfer of virus, mostly because the virus is dead. So the virus can enter the cell but can never exit. Okay. But also, and I didn't have time to show this data, we have generated cells that we use in CRISPR-Cas, where we depleted RAP27A and B. Yeah. And so those cells, so those two molecules are very important in the formation of exosomes. So when we deplete them, there is no exosome formation. So we have tested all the same data that we show today for this virus as well as for other exomir viruses in those systems, and the effect is gone. So that is another way of controlling, as you mentioned.
Okay, great. Thank you.
Thank you for your question.
Thanks, Carolina. So I see that we have about 2 minutes left. Before we formally end this, I'd just like to thank you and Eva and Avira for presenting. I think it was a fantastic synthesis of, like I said, a lot of work. So in about 2 minutes, we'll formally close. There are quite a few questions left. Do you have a few minutes or do you have a hard stop at 3?
I can answer all the questions.
We'll be here all day, but hopefully not. Okay, wonderful. So with that being said, anyone who does have to duck out, thank you guys for attending. There will be a survey that pops up on your screen. And as always, we do appreciate that you answer those just to help formalize next